Kenko Hacks, a podcast about wellness, health, and biohacking.
Or lifespan.
Yes.
So most people don't think about it at all, and they just eat like it's their last meal, and when they die when they, so that's not good enough for me. And it may not be 15 years. It may be 5 years. It may be 50 years. I mean, that's, but the challenge and the fun is, well, I guess we'll get into this, right? But by knowing that aging is a biochemical process and disease is a biochemical process, it doesn't have to be where, now you have disease, now you start paying attention to your biomarkers. We can do this while we're healthy and potentially delay disease and optimize lifespan, right? The question is how much, right? So if I was going to die at 50 of a heart attack, because of my genetic background, maybe I get to 60 or 70. So, and the quality of life should be better, right? So, I'm different, significantly different from all the other anti-aging people out there, because it's all just, eat this way and you'll be this healthy, or take this supplement. But where's the actual data? Where's the actual data that it works, right? So,
yes, absolutely. You have opened my eyes twice. So once, you are so I might have mentioned it in my email, but I have been taking blood tests, annual tests. Now I have changed it to every quarter of the year, influenced by you, definitely. But with a very vague general understanding that they are useful, but I never, so that's because you taught me the values, or what the optimal values are, basically. I had no, I really just, and it's so complex, right? Especially for things like cholesterol, there are optimal values actually for different age groups as well. So, and as you are a scientist and you are such a data cruncher, and you are also such a brilliant presentator, I think your latest video, selenium, and the values come from so many different places, but you do this side-by-side presentation of nutritional study, and you show besides that, you show, however, the blood circulating selenium, and then I think it really speaks to regular people like myself, who are non-scientists, to understand. And then I really immediately measured my Brazil nuts afterwards, and I'm never going to munch them mindlessly again. It's really the message. I'm not saying maybe those messages weren't there, because information is all around us today. But your videos truly speak to the audiences, I think. And
So I hate it when, I'm classically educated. I have a PhD and all that, right? But I like to think I'm from the Elon Musk, the Steve Jobs, school of learning, where you don't need to have advanced education to be quote unquote smart. And I try, even though I am classically educated like that, I wonder if I could have. Well, I wouldn't have been able to get to this level without the classical education. But what I know in my approach, anybody can do. I mean, I'm literally driving to get my blood test done, and I'm tracking my own diet. I'm using simple tools. I'm not doing anything advanced. Now, at some point, we'll get some machine learning and some AI into, instead of me going to the correlations that I show, there's going to be more advanced tools to do that. And I just put my data in, and it gives me the exact diet recommendations. And just like a Tesla car, where they have billions of miles of data to teach it to drive itself, eventually I see this approach going there. But I hate it when scientists speak with jargon. It's funny too, because, I try to make it as easy to understand. And for some people, they say, it's still over my head. And I tell the truth. I'm like, look, when I think things are over my head, I read it again. If I have to read it 6 times, I read it 6 times. I listen to it over and over. I look things up, So, I try to make it as easy to understand as possible. But then it's interesting because I'll be on like calls with, work colleagues. And, but it's funny because like most people don't have the attention span to listen or watch for hours. I mean, for example, like the average watch time of a YouTube video is 4 minutes and 20 seconds. So if you post an hour-long video, maybe you get 6 minutes. So knowing that, I try to keep it straight to the point, as you know, no extra words, just straight to the point. I don't even edit it. I just go in. Sometimes you hear me say, "Oh, I want it to be real and authentic," right? So, but the long story short that I'm trying to say is that, you know, it's taught me also about human behavior, because knowing that most people don't want to listen or watch for extended periods of time, it takes a special mind. Like, I'll sit there and watch a 10-minute video. I don't have a problem because I'm interested, you know, but for the average person, knowing that they have a shorter attention span, even when they're interested in it, I'll hear people talk. They'll be asked a question and they'll go into and it's a simple question. Whereas, you know, we're having a conversation. It's different, right? But something at work, you know, as a scientist, they'll get asked a simple question, and it's like I've almost Twitterified my responses to where I'll give, you know, like a 30-second answer, because I know most people don't have the attention span. So, you know, I'm just like the regular person in that way. I don't want people to talk to me for hours. Get straight to the point. Get straight to the data. That's what it is, right?
Yeah. Definitely, I think that this spirit, just which you just described, straight to the point. It's definitely I can sense that in all of your videos and other interviews as well. Just to give a little bit of structure to our interview today, and I very much wish that this interview will reach audiences who haven't yet discovered your videos, and so. Could you please maybe introduce your YouTube videos? So we already started talking about the details and latest video on selenium, but would you maybe like to introduce them, what they are about, and what your goals are with them?
Yeah. So my interest is in optimal health and longevity. You know, I can't stand the idea of getting older, whether it's you know, externally in terms of my fitness or internally in terms of my organ health, right? So for me, as I mentioned, aging is a biochemical process. It's not something that just happens 50 years from now. It's something that happens for every day during those 50 years or longer, right? So, so with that in mind, you can track all these things, and then by intervening when things aren't optimal, you can potentially optimize them in youth and keep them there for longer, delaying aging, right? So, the concept with the videos is to basically dissect bit by bit the biomarkers or other aspects of aging that maybe people don't know about. Or, you know, I've seen other channels where they'll say, oh, we're going to do a paper review, and then they'll go through the paper. But the fact is that paper has data from a multitude of other papers, you know, that science isn't just one paper. It's built on the foundation of other work. And I try to give as much context as possible, you know, so that it isn't just one story I'm telling. It's within a broader context for understanding. So, you know, I try to focus. I'm kind of like a biohacker, aging news, but also mixed together, you know, and you know, not like this biohacker stuff where I'm going to talk about red light therapy or blue light therapy or EMF, like these things that are biohacker fringe in my eyes, but seem to be very popular. You know, I try to focus on the tried and true, and you know, so things that can help you in terms of diet, things that can help you in terms of blood testing, you know, education in terms of the hottest trends in aging, and trying to make it as easily digestible, you know, to the broader public. I plan on doing this, you know, making these videos for as long as I live. So, I mean, you know, it's only a matter of time. And it's interesting too, just to point out, you know, I could get the sense that your passion for aging and longevity in the email, and plus we're in a couple of groups together, right? So I don't actually do every podcast that's asked of me. So you know, the audience size that this reaches for now, I'm not even worried about it. Knowing that you have a passion for this and the people that listen to you have a passion for this, if that's 10 or 50 or 2,000 or more, as big as it is, you know, so.
Yeah, absolutely. But so I already started talking about that, but so you opened my eyes twice on your videos. So first, you so elegantly and meticulously many times show what the optimal values are for different blood markers. And even though I have been taking different blood tests for over 10 years, I had no idea how to, because you see, unfortunately, people see the reference ranges, right? And then, you know, usually you are within reference ranges, and then you feel like you're fine. Would you like to explain the differences between optimal values and the reference values and why one should focus on the former one?
Yeah, so the reference range is a population-based average, and it isn't intended, so if the population has certain values, you're within the 5% to the 95th percent range, and then if you're beyond to the left side of that distribution or to the far right of that distribution, it's considered abnormal or out of range. But generally, those reference ranges aren't intended for disease risk or even longevity. So, and the reference range doesn't tell you, how does it change with aging? Like if you see your values going up or down, it doesn't tell you, is that the right direction or the wrong direction? And the common thought, for people who get their blood tests, however often, once a year, twice a year, is, oh, my blood tests are right in the middle of the range, so I'm good. That's how I used to think about it. And I've heard others say it like that. But the fact is, for most biomarkers, that isn't true. So from my perspective, knowing how each of the biomarkers that are on just the typical blood test that you get from your doctor every year, the standard chemistry panel and the complete blood count, which are reflective of many organ systems, the health and functioning of many organ systems, and that these biomarkers have been studied for 50 to 100 years plus, depending on the biomarker. Some of these biomarkers have been studied for 50 years, some more than 100 years, like glucose. So there's a wealth of data. There's a wealth of data in terms of how they relate to disease risk and how they change during aging. Whereas in contrast, not that I'm, not to knock it, but think more recent things like the epigenetic clocks, which are also a measure of biological age, are more new. And there's much less data on what aspects of aging they're actually measuring, and then even randomized controlled trials to even optimize these things. So they're going to be even bigger than they are now. But so that's why I focus on the things that are tried and true. It's not the most cutting edge, knowing that something's been around for 100 years, but there's a wealth of data. So knowing that and doing as comprehensive literature reviews as I can, which means that, if I look up a given biomarker like albumin, I'm not just getting to the first paper I find and then, saying, well, that fits my bias, so I'm going to stick with that data. Like I look up literally all of the data, albumin and aging, and I see what the consensus data is even in the largest studies, because another variable is if I have a study that only looked at 200 subjects and I have a study that looked at 1 million subjects, which is the truth, right? So there's a little bit more to that, knowing that smaller studies can lead to either accurate results or inaccurate results, depending on the population. Whereas if you have larger groups, very large groups, now you should have a more reflective part of the story. So just going back to albumin, as an example, so I did comprehensive reviews how these biomarkers change during aging, and then also comprehensive reviews on how they relate to disease risk, and not just focusing on things like one disease at a time, which I've seen a lot of people do. Oh, this is associated with heart disease. Well, you may take a statin for your heart disease, but what's the risk for death from all causes? Is that because if that goes up and your heart disease risk goes down, is it a net positive or negative? So I focus on all-cause mortality because that's risk of death for all causes, right? So then putting those two pieces together, it should tell you now within the reference range, do you want your values to be high? Do you want them to be lower? Knowing how it changes with aging and disease risk. So with that in mind, then the fun part becomes, okay, I have this value for this biomarker and it's higher than it should be based on the Lustgarten quote unquote optimal range. How do I change that? For the common ones like, glucose or inflammation, those may be more relatively easy to change. But if I say mean corpuscular volume, the MCV, first of all, most people
hard. That's very hard.
It may not be though. That's the thing.
Yeah.
But when, for starters, most people would hear that and they'd be like, "What even is that? I didn't even know it was on my test." So with the videos, I try to give a brief introduction into what it is, and then the aging and the disease risk. So the fun part then is figuring out which aspects of your lifestyle, whether it's diet, exercise, supplements, sun, whatever it may be, vitamin D, that impacts the biomarkers, and that takes time. So there are companies that promise, whether it's based on a blood test or a gut microbiome sample or whatever it may be, that'll say, we're going to sample your data and give you precise dietary recommendations off one blood test or one stool sample, which from my experience is ridiculous. I mean, this takes time. you need to have a lot of your own. Like, imagine if you had a scientific study and you said, we did one data point and we accurately, in one data point in one person, the study's underpowered and undersized. It wouldn't fly. So you need a lot of data,
No.
a lot of measurements, and rigorous trial and error to accurately get close to what the truth may be. So yeah, that's my approach in a nutshell.
Absolutely. And so you have opened my eyes twice. So first, the meticulous, and for I would say that the data, as you say, there are large data on different blood markers already. But as a non-scientist, I can, of course, today I can Google PubMed and find thousands of, but crunching data, extracting the vital information, even so converting from, that's a very banal example, from millimole to milligram per deciliter, for example, it's just so error prone and not difficult. As that, it's not a rocket science. There is a way to do that, but for most, I think, majority of people like biohackers who are non-scientists, it's just too tedious, or not just becomes impossible practically. And that's where I see your videos giving such great values. You really crunch those data for us and present them in such a beautiful, elegant way. And I cannot recommend them highly enough. They will, of course, be linked in the show notes. But as a second, so eye-opening experience with your videos. So you basically optimize. You are trying to optimize your blood markers through nutritional strategies, and a lot of vegetables are involved in that. And first time, I have to say, it was one of your videos, one of the first your videos I watched. You presented your dietary vegetable intake, and honestly, I thought there was one extra zero, and quickly I noticed you don't do such a mistake very often. So, but it was up to 2 kilograms of vegetables per day, which is respectable to say the least. And that has definitely changed. So that was a couple months ago. I watched that video, and it completely changed my dietary. So of course, I have always known, as person interested in health, diet and exercise. I had heard it maybe a million times, but you really gave me the concrete idea what it really means, and you back it up with your blood testing. And I think that's a very unique thing about your videos. So would you like to maybe explain for the audiences what your dietary strategies are?
Yeah. So in terms of your two eye-opening points, just to go through that. So I guess in my earlier videos, I was focusing all on the Western metrics, the Western units of measure, in reporting all of the data in milligrams per deciliter. But it's only in my more recent videos that I've tried to include both data, or in more of my more recent videos. But that's definitely a focus of mine too. Because I have broad appeal to an international community. I have to make sure that my data is not just in pounds, but also kilograms and millimoles and milligrams per deciliter. So I'm not perfect with that, but literally every video you see from now on will have both units of measure. So, and then in terms of the diet and exercise aspect, so. It isn't just, so I don't just use diet to optimize at all. It's a big part of what I do, but exercise and how often, for how long, and at what intensity. So I completely changed my exercise approach based on, again, data, right? So I track my own fitness. I won't say who they are because I'm not sponsored by them, but so they give me data like resting heart rate and heart rate variability. And anyway, those things change with age too. So I have some videos on that. But before I was tracking, or when I first started tracking these things, I noticed that my data were not as good as they could be. And that was surprising to me because, I did cardio every week. I mean, walking may not seem like a lot for most people, but 15 to 20 miles a week is a lot of walking. That's a lot of cardiovascular exercise. And plus I was strength training, probably 2 days a week, but up to 3 hours a session. I for whatever reason, I have gorilla genes. And I like throwing heavy stuff around for a long period of time, or I liked. But it would take me a lot of days to recover because it was for so long and lifting so much and doing so many sets and reps. So I completely changed my program based on trying to get my data for resting heart rate and heart rate variability to closer to more youthful levels. So And even that, it's still a trial and error process in terms of, okay, I did this long of a workout, and I know that if I do this much tomorrow, I can't do this much the next day. And one reason for that, goes back to the lifespan story, which is exercise is well known to increase average lifespan, but it doesn't increase maximal lifespan in animal models. And in further support of that, where are the bodybuilders, whether they're just lean and ripped or humongous, big bodybuilder types, or the marathon runners that are 125 years old? Like, it's the exception rather than the rule where you see a marathon runner that gets to 100 years old. So, there's definitely something about exercise that increases average lifespan and health span and fitness for longer. But what about maximal? So, by tracking these metrics, at least heart rate variability and resting heart rate, I can try to maybe gain the maximal benefit from exercise, where I'm not just getting a boost in average lifespan, but potentially getting me closer to the maximal. So, yeah. And then what was you asked the question about diet in terms of,
yeah, so you always emphasize, so what sticks out in my eyes when I listen to your old videos, so you always emphasize the nutrition dense food, so nutrition dense per calorie.
Yep.
And that's, I think, very worse for people to listen to.
So that came from a few different angles. So I basically literally had no idea how to eat when I first started this journey. I tried to minimize junk food, way back when, and eat a healthy diet. But it wasn't until I started regular blood testing, that I started to try different things. So around 2015, 2014, I actually tried going, as close to raw vegan as possible, because there was a lot of stuff online and a lot of published studies showing that, vegans are healthier and leaner and all these things. But
yes.
So I actually tried going pretty close to raw vegan, and that didn't optimize all of my biomarkers. I mean, things like triglycerides went up twice as much as my normal, and my HDL actually went down to levels that are 28, which in hindsight, I mean, it seems outrageous. I mean, that's
not good.
Yeah, not good, especially knowing that the optimal range based on the data that I've put in the videos is somewhere in the 50 to 60 range for a man and higher for a woman. So, I've tried different diets, trying to find the path that is best for me, right? And it may be different for everyone, but in terms of the vegetable story, it wasn't until I started, literally mega dosing my vegetable intake, that I started to see lots of favorable changes. Like, just as one example, I have a kidney function video where, the strongest correlation with better kidney function for me is the quantity of my vegetable intake. Now, I've actually cut. But then again, I'm not married to these ideas, So whereas 100 grams of fiber, and mostly from vegetables, broccoli, cauliflower, carrots, I mean, eating, a couple kilograms or more of that a day, is correlated with better kidney function in my data. I've actually cut my fiber intake by about 20 or so grams, which doesn't seem like much, but that's a big difference for me. I mean, it's a 20% cut. Which
yes.
the reason, but that's a temporary thing. I've done that before. And one reason I did that is because, so I have hypothyroidism, and I've been taking thyroid medication for the past 25 years. I went to a endocrinologist. They did the sonogram. I have a atrophied thyroid gland. And one reason is probably because I lived pretty much off junk food as a kid, but was active. So I was relatively lean, but I was probably killing myself metabolically. So, anyway, the long story short is there are foods that can affect the thyroid, like cruciferous vegetables, broccoli, cauliflower, Brussels sprouts, etc. So, broccoli and the cruciferous vegetables are all stars for health, whether it's nutrient density or fiber content, even protein. They're fantastic, especially if you're eating big volumes of it. So I started to notice that my heart rate variability, which had been in the 50 to 60 range, and actually closer to 60 for a few months, had started to dip into the 40s to 50s, and that seemed to correlate with me going to, eating about a kilogram combined of broccoli and cauliflower every day, and I'd wake up in the mornings feeling horrible, as if I did workouts every day, even on rest days. So I've cut doing another experiment. I've cut my cauliflower and broccoli intake completely out, still getting about 80 grams of fiber a day from the rest of my stuff that I'm eating. But my heart rate variability is now
Yeah.
back up into the mid 50s. And this time of the year is actually rough for me because I don't know if you can see, but my eyes a little bit bloodshot. So I seasonal, maybe
Yeah, I
not
can see the red. Yes.
red. Yeah. Seasonal
Yeah.
allergies,
Yeah.
kill pollen and grass kill me every year. So, and this is actually my worst data in the summer months for my biological age data with the blood test. Inflammation goes up, eosinophils go up, things going in the wrong direction. And one reason is because of the inflammation. Anyway, so considering that I know, so anyway, that's my most recent experiment, is with cutting my vegetable intake just by a little bit. So now there's a balance between if my heart rate variability is now going closer to a 20-year-old because certain foods may be adversely affecting, the parasympathetic sympathetic balance, which is HRV. But knowing that eating 2 kilograms of vegetables, including broccoli and cauliflower, maximizes my kidney function and gets it going in the right direction and not going in the wrong direction. So now we come up with, well, you improve one thing and maybe make something else worse. So it's trying to figure out how can I work around that while still optimizing the whole system. So it's not a perfect system. I'm always doing experiments, but with all this data, like I said, eventually you get closer to the truth. And then okay, if I eat this much broccoli, it'll negatively affect this. Or this much broccoli, it won't negatively affect that. So you can get closer to this idea of true personalized nutrition, knowing how much is truly optimal for you. So, but vegetable. So just to finish the vegetable story.
Yeah, that's not easy. Yeah,
I know.
Actually, yeah, please.
Yeah. So another aspect of the vegetable story is most people don't eat enough fiber, and what I mean by that is evolutionarily, there's published evidence that the Paleolithic man, and I'm by no means a paleo type where I'm only going to eat a paleo diet. I'm not that at all. I follow my biomarkers. That's a completely, if you say, are you paleo, keto, vegan, vegetarian? None of the above. What my blood biomarkers say, that's what I follow. That's the diet that's best for me. So, but anyway, so evolutionarily, there's evidence that the Paleolithic period, which is a 3 million year period in human evolution, for the majority of that, the prehistoric people ate meat for sure, but a humongous volume of plants. In fact, on a 3,000 calorie diet, the average fiber intake has been estimated to be about 100 grams per day. So when you do crunch the numbers, 100 grams per day on 3,000 calories, that's 3.3 grams of fiber per 100 calories. Whereas most people today average at most 1 gram per 100 calories. So 2,000 calorie diet, 20 grams of fiber, whereas based on evolutionarily, we evolved eating probably closer to 60 grams on a 2,000 calorie diet. So, there's also evidence too that if you can, and this is in flies, if you can put the flies closer to their more evolutionarily accurate diet than a lab diet, they live a little bit longer. So I started with that philosophy, and then actually the data verified it, in terms of the blood test data. So I started with the published data, because there's actually some value in that too, where, you'll have, all right, this study suggests this, should I do that in my own diet? And then you don't measure biomarkers, right? Which I did that in the beginning. But then I realized my biomarkers actually have the truth, right? If it works in a big study and I try it and it doesn't work on me, well, what do I need it for? It doesn't work, right? So the vegetable story goes back first to the evolutionary story. And then I saw more positive than negative changes in my data, because that's the key. Like, if you only look at glucose levels, but it messes up 6 other things, and don't look at those 6 other things, you'll think it's net positive. But so I try to look at as many variables as possible. And then, is it a net positive or a net negative or neutral, right? If it's neutral, you can keep it in. So the other aspect that I'm getting to is the fiber story. And this likely ties in with the evolutionary history story, but if you're only eating 20 grams of fiber per day, you're neglecting the impact of fiber on the gut microbiome's ability to produce metabolites that can not only optimize intestinal function, but that have been shown to impact the brain, muscle, liver, every organ in the body. So most people don't eat enough fiber to basically activate those mechanisms. So for me, that's what I do. I study them in part. I study the gut microbiome and its metabolites as my day job, right, as a scientist. And knowing the published literature on the effects for these super high fiber diets, which are rarely used in studies. So that's the other aspect of it. And the only way to get there, the opposite view is people say, well, just supplement with resistant starch or this fiber or that fiber. But that goes back to the supplement story. If you're megadosing things that we didn't evolve to get in megadoses, is it going to be truly beneficial for you? So the lowest risk strategy is to get it how we evolved to get it, which is by eating a ton of plants. And again, that doesn't mean being vegan, but a ton of plants, as many as you can fit in, and that doesn't cause GI distress, that doesn't make you feel terrible in the morning, and that optimizes your biomarkers. So that's the reason for the abundance of vegetables. Now, that also said too, I'm not saying this approach will work for everyone, right? At the end of the day, if it improves more than not, then go for it. But if you show in your data that it doesn't improve more than not, then you've got to find what works for you. For me, it seems to be a net beneficial thing. And as a last point too, I'm genetically a volume eater, which means large volumes of food satiate me. If you put me on a 2,000 calorie diet and it's on pizza and ice cream, which is high calorie density but low nutrition, I may feel great for 5 minutes, but then in 2 hours, I'm gonna be like, I want to eat more, right? Because it's a small volume of food. Whereas if I eat a pound of carrots, I mean,
Yes.
it satiates me for a while, but that's just how I'm built. Like, unfortunately back in the day, and, it's like a recovering junk foodaholic. As I mentioned, my childhood, I ate so much junk. I'd eat whole cakes, 2,000
Yes.
calories, so I've had days.
Wow.
And yeah, well, that's not even that much. I've had days, 20 years ago or so, where I could easily take in 10,000 calories in a day, eating a whole gallon of ice cream, a whole pizza, etc. So, but learning about that and going through the years, I know I'm a volume eater, and vegetables basically satisfy that, so that I can, yeah. But that said, there are also days when I'll eat junk, but I try to make it an infrequent occurrence rather than, an addiction.
Yeah, but that's the genes you have, the big eater gene. It's a quite a big advantage for you now, because I don't have the gene, and I actually struggle to eat a lot of vegetables, just because I don't have that gene. So now that you have changed the dietary habits from junk food to a lot of vegetables, I definitely see it as a huge advantage. But there are a couple of questions around that. So you mentioned the balance. So if you 6 biomarkers goes to the right direction, 2 down to the wrong direction, it's still, that's because there's always balance. You have made many videos where you show that, well, C-reactive protein goes the right direction, but glucose goes to the, and
Yes.
it's very intricate, and it's the complexity. And one area which I wonder how you manage is minerals, because there are so many of them. And selenium, we discussed your latest video, and that's really great video, because I really learned the selenium, the 300 microgram, and that's very, very useful and important. But there are so many others, and I think many others don't have even daily recommended allowance values, even though they might not be so useful to begin with. But like boron, for example, and so many others. How do you manage minerals?
Yeah, so that's a great question. So that goes to the issue of, small scale versus big scale. So small scale, it's very easy to have reductionist science and say, I need to optimize every one of my vitamins and minerals in blood. So actually, let me take a step back. So first, I start by optimizing everything dietarily in terms of at least the RDA. So I log my food. I'm sure I log my food every day. I weigh all my food. I enter all that data every day. I've done that for 6 years. And where some people think that's, crazy and whatever, I mean, that's just the background, right? You hear police in the background.
That's no problem.
So I've logged my food for 6 years, and where some people may think that's crazy, I mean, that's just how I'm wired. So I weigh it all. I put it into the, Chronometer, not sponsored, at least not yet. And that gives me my vitamins, minerals, and macros. And then I put that into an Excel file. And then I take the average amount during that period and correlate it with my blood test. So at least in Chronometer, I shoot for the RDA. But then there are certain nutrients that I've done, or for most of them, I've done deep literature dives, and that just the RDA isn't optimal. But this goes to your question of how do you optimize the minerals and vitamins. And, so first it starts with diet. Then the second question is, if I measure all of the vitamins and minerals in blood, how much of an impact will that have on my health? And so then it goes to the biomarkers, right? So to be honest, I haven't spent a lot of time. I haven't spent a lot of time on quantifying my blood levels of micronutrients. Not yet. That said, my next blood test is on Monday, the third one this year.
Yep.
And
exciting.
Yeah, for sure, right? It's going to be my worst. I expect the worst data because I know my data this time of the year. But selenium is that I'm going to measure my blood levels of selenium, right, and see how close it is to that 166 micromolar that was associated with in that meta-analysis. So, and then if it's way too high, I'm probably going to cut my diet intake a little bit to actually see if I can optimize that better. But it goes to the bigger picture, and this is what I mean about the small scale versus the big scale. If I'm obviously going to measure my other, those other 40 biomarkers on the blood test. So, and then quantify my biological age using Levine's test and then aging.ai. And then knowing that how, I'm usually 12 to 14 years younger using Levine's test, and then aging.ai, I'm somewhere around 30, which is around 20 years younger. So if I get a blood selenium value that's extremely low, right? But the comprehensive set of my big picture biomarkers are youthful, I probably won't change my selenium intake to change my blood values, right? And that may be what blood values would be optimal for me. Now, conversely, if my blood levels of selenium are way too high, maybe they're twice as high as they should be, I'm probably going to cut down on my dietary intake. But then the net effect is the big picture and looking at all of the blood biomarkers and the biological age metrics. So I see a lot of people, and actually other MDs in this space, where they'll do full vitamin mineral profiles on their clients. And for me, focusing on the bigger picture and all of the other well-studied biomarkers that have way more data than 6 studies, for example, on selenium for the blood levels. Focusing on the big picture is how I would address that. Try not to get lost in the weeds with the blood levels of the vitamins and minerals. Start with the diet optimization, but then make sure all of the big picture biomarkers are optimal. And if they're not, then trying to tweak, right? But that goes into too. Yeah, I focus on the diet and, even quote unquote mega dosing some levels of some nutrients that, based on the RDA, in my eyes, are grossly deficient. Like vitamin K, the adequate intake is 100 micrograms. There's published data. I have a vitamin K video that at least 1,000 micrograms per day may be optimal. And actually, there's scientists at Tufts who spent their whole life focusing on vitamin K and have actually raised the issue. Based on these studies, shouldn't it be higher? And I don't want to wait another 20 years for the RDA to be updated before they realize, I see the data now. I'm making basically a leap, right? I'm making an educated guess that based on these studies, it's not optimal. So, but the net effect of the biomarkers, right? If my biomarkers were all messed up and, all these dietary recommendations that I have for me and potentially for others, if my biomarkers weren't suggestive of youth, then I'd be on the wrong track.
Yeah. That's a really fascinating answer. Another question I have on this around this nutrition topic is, so when I hear nutrition dense food, before watching your videos, I always used to think like organ meats, like liver. What's your take on organ meats?
Yeah. So within the content, for me, it's show me the data, and it's if someone wants to eat nose to tail, whether it's eating organ meats or just meat, if you can show me your comprehensive set of biomarkers, not just selective things like my triglyceride to HDL ratio or glucose, which is what, from my experience, many in that community do. If your comprehensive set of biomarkers are going in the right direction and you have a biological age that's far younger than your chronological age, then go for it. Now, beyond that, the question could be, are organ meats a part of a healthy diet? Yeah, they could be. But then again, it comes down to individuality. For me, I'm not a fan of, the idea of eating liver is disgusting to me. That's just me. For others, have at it. If you enjoy eating organ meats, have at it, But I think it should come down to the biomarkers at the end of the day. For example, Paul Saladino, who's an MD, and he was on Joe Rogan's podcast, he had been notorious for being exclusively carnivore. And he posted his blood test data on in the show notes. So I made a video on that, what's his biological age? And if that diet is as good for him as it should be, you'd predict a very low biological age relative to his chronological. So I think he was only like 3 or 4 years younger than his chronological age, which, I mean, I'm consistently
Yeah.
12 years. And even at my worst, 9 to 10 years. I've actually tweeted it at him, you can improve your biological age. Maybe you can add some other things into your diet that can improve some of your biomarkers. And now you're not eating it with a dietary ideology. You're eating based on what is optimal for you. So since then, I don't know if it's because of me or others or his own ideas, whatever it may be, he seems to have expanded his perspective, and he's a big nose to tail guy. So I don't know what his biological age data is since then. But just all foods can be included as far as I can tell, as long as your systemic biomarkers are net positive, mostly going in the right direction or indicative of youth. But even then, one blood test isn't going to do it. Like if I added liver into my diet, my biological age was 30, and now I have liver in my diet. Now it's 25. That's a big jump. So is that a real effect? I'd have to do the experiment again. So I keep liver in my diet. Whatever else I took out could also be contributing, right? What if I took out 500 calories of junk food and liver was replacing junk food? So then I would know liver is net beneficial compared to, what if I took out things that I know benefit me positively, like vegetables? But let's play along with that story. So now we did one test. On the second test, we'd have to see, is that a real effect? Okay, so now we get another 5-year reduction. So then that 2 data points isn't enough either. You'd have to see, is this effect going to be associated with slower biological aging? That's how I would look at it. So one blood test wouldn't be enough. In my eyes, you'd have to do the blood test for a few measurements to see, is it consistently related with a multitude of better biomarkers? And then is the age-related change going to be better than what it replaced? So that's how I would see that. For me, not a fan, but for other people, if they want to go for it, enjoy.
Yeah, I understand. Very rational approach, as follow your videos as well. You mentioned previously that it's not all just nutritional strategies, other components like exercise and also frequency and so on. Talking about frequency, so many people are into fasting, right, these days, and they do that often because of the assumed benefit for mitophagy. So you accelerate mitophagy and mitochondrial renewal. Do you have any nutritional or any strategy for optimizing mitochondrial health?
Yeah. So probably the best intervention for mitochondrial health is going to be aerobic exercise, because you're actively using your mitochondria. I'd bet that. So now nobody's compared diet versus regular consistent cardiovascular exercise to see which one best improves mitochondrial health. But I find it hard to believe that diet would be better than an approach that actively uses mitochondria while you're doing it. I mean, if you're going for a walk or for a long walk, or you're running on the treadmill for longer than 3 minutes, you're engaging aerobic metabolism, which is directly using your mitochondria as much as possible. So exercise would probably be the best approach for that. But in terms of fasting, I mean, there are other health benefits besides its potential effect on mitochondria. It's the duration of the fast. That can be important, but then also your background calorie intake. Because if I eat a large pizza and some other junk in the morning, but then fast until the next day, I'm at a caloric excess. So even though I fasted, it's probably going to be net negative, just because not the fasting part. The fasting part obviously is going to be good, but still I'm in a caloric excess for those 24 hours. So I'd probably need an even longer fast to get me to where now I'm at maintenance or a little bit below if you're trying to, get leaner. So with that said, I do incorporate fasting. It's a big part of my approach for a couple of reasons. One is I used to have terrible sleep quality, and I shouldn't say I used to. I'm constantly trying to work on optimizing my sleep quality and quantity, which for me, they're both related. But so I used to fast all day, literally not eat anything in the morning, drink some tea, go to work, come home between 5 and 6, and then eat, my daily caloric intake somewhere in the 2,500 calories range from 6 to 9 at night. And then I'd wake up 2, 3, 4 times in the night because vegetables are filled with water. There's no way to get rid of that. It's, you got to wake. So I'd wake up so many times during the night and feel terrible in the morning as if I didn't sleep. So I completely changed that. and flipped it around to where now, and actually seeing some of the time restricted feeding studies, some of that positive metabolic data, switched it to where I eat the majority of my calories before 3 o'clock. And in fact, some days 1 o'clock. And if I do eat something, maybe it's, you know, 100 calories or two, but nothing significant to impact, you know, or impact as much, waking up too many times during the night. And it's funny because when I'll eat like a giant salad, like a pound of romaine lettuce, half a pound of spinach, and then some other stuff, which is a lot of water. Even if I do that at 1 PM, I find that I still have to wake up during the night to use the bathroom, which that's like, 8 hours before bedtime. So, and then I'm sure people will chime in and say, well, you may have prostate issues and all this other stuff, but I eat so many vegetables and fruits that I'm chronically overhydrated. So, but anyway, that said, I try my best to fast for that, 7 or 8 hour period before bed, or eat as little as possible, because for me that optimizes sleep quality. Now, whether that approach optimizes my blood biomarkers, I'm not sure, because even though there is some favorable metabolic data for every other day fasting or 1 meal a day, with mice living longer on 1 meal a day, I don't see a dramatic difference in my biomarkers, before I change to eating mostly all my calories in the morning versus more at night. But even from just the sleep quality and quantity, I see that there's a tremendous difference there. Right. The big.
Why do you mention about this? Yeah, the hydrating effect of vegetables, I can attest to that personally, because before switching to this quite a lot of plant-based diet influenced by you, I was trying to, for one marker, not blood marker, but we have a weight which gives digital scale, which gives you not only your weight but also water percentage, so fat percentage, and so on. And previously, I was trying to, because your body water percentage goes down with aging, and I was trying to get it up just as a personal interest. And I couldn't. I failed for a couple months actually. So of course I tried to drink water, a lot of water first, but it doesn't help actually. It really just doesn't help. And I almost gave up it because nothing happened. My water concentration was like in the low 50s, and I wanted to give it like to the mid 50 or closer to 60. So, and as soon as I started eating more vegetables, now it's around 56% of my weight. Wow. So it makes quite a lot of difference. I can definitely say that vegetables hydrate, and yeah.
So that's interesting because even though obviously vegetables have a very high water content, just drinking water should have the same effect on the body. So if you drink water, you're going to absorb it. Potentially you'll hydrate more systemically, right? But when you eat vegetables, in my eyes, you should have the same effect. You eat the food, your body breaks down all of the nutrients, absorbs them, but then also absorbs the water that are in those nutrients. So to me, then that means you're getting more water into your cells because of vegetables just versus only water. That's very interesting. But I was going to take it to a different point of view. So the age-related decline for bodily hydration is more related to muscle mass than it is to drinking water. So our muscle cells are hold most of the water in the body, or hold a great amount, 30% or so. 30% of the body weight is muscle mass, or it can range. So if there's an age-related decline in muscle mass, that's a major tissue for storage of water. So for most, the age-related decline for muscle mass is well known. So if you have less muscle mass, you'll be storing less water. You'll look like you're more, quote unquote, dehydrated. So I'd actually focus on that aspect of it too, making sure that your strength training approach, or how often you exercise, all that stuff. So you're trying to keep your muscle mass at least the same, or try to improve it, because if you're not trying to improve, maybe you stay the same, right? So I'd look at it. I'm glad that vegetables have contributed, but that's a big part of my approach. I don't want to have any less muscle today than 50 years from now. So making sure I stay, strong and fit. And actually, I should have some. It's easy for me to make videos where I'm not, showing my face or my physique, because it seems like when I just present data, people will argue with me about the data if they have other data, which is fine. I don't have a problem. But the second you start posting physique updates, now you get, all kinds of like, you could lose a little bit more fat here, there. And I don't know. But the fitness
But you
approach.
post your blood markers, so.
I have posted some fitness update pics, and I do have some pull-up videos, but that's on my
Yeah, I
to-do
have
list
seen
for the
that too.
summers. Yep. But so I was doing 12 a year and a half ago. It's time for an update. Am I still doing 12 pull-ups, and full, because I see people ego lifting and they're just doing quick pull-ups and they barely get their chin above the bar, so. But the long story short is, if my strength is the same today as it was a year and a half ago, how much muscle mass have I lost? And if I have lost muscle mass during that period, well, my strength has stayed the same. So, now I have more functional tissue with that. So
Right.
it's a big part of my approach, making sure to maintain, or, at worst, maintain muscle mass as a part of the hydration story during aging.
Yep. That's very interesting conversation too. I wish to touch upon your book, Microbial Burden, as well. We already discussed the fibers, and so your YouTube videos. I watch them many times, and I look forward to going back to many of them in the future as well, because every time I learn new things. Your book, I have learned. This is my first cycle of reading, so I know that. It's just so dense, information dense, that's so much to extract. But one question I got when during this first through reading for me, so microbial burden, is it so that everything which comes from, which are related to microbes, which are found in our bloodstream or organs or wrong places in our body, are they considered the microbial burdens?
Yeah. So, but more specifically, they shouldn't be in those tissues. They shouldn't be in those places. Like the idea that microbes are in the blood. There are studies, a handful or a few dozen that exist on that. But it's been long thought that the blood is sterile. And a big reason I wrote this book is most people think exactly like that. Like, okay, I have microbes on my skin or microbes in my mouth or my gut. But there's actual data that they're in other places that they shouldn't, and that they're in other places that they shouldn't be. That there's evidence suggesting that they may be impacting not just diseases, but also aging itself. So that's the major reason I wrote the book, to expose that idea to the world, and also based on the published evidence that those things. But yeah, when I mention microbial burden, I don't just mention the 99% of our microbiome, our whole body microbiome that's only in the gut. I mean, they're in places that they shouldn't be, and they grow in number in places that they shouldn't be during aging and with disease. So yeah, it's a cumulative burden everywhere.
Yeah, but my question was maybe more specifically: are they any microbes in the wrong places are considered burden? Not only like bad microbes, so because of course maybe it's scientifically there are no bad or good microbes. That's of course point of debate. But I was just wondering if it's about the specific species, or the
Yeah.
places they
Yeah.
exist.
Great question. So this is the cutting edge of knowledge. We're at the cutting edge of knowledge right now. So there's a little bit of speculation and there's a little bit known. So, for example, there is a blood microbiome. Even in youth, there are microbes that are found in the blood. Like if you do genetic analysis of the blood, and you can assess which types of microbes, whether it's bacteria, fungi, or virus, they are in the blood, even in youth. So then the composition of that microbiome, just using the blood as an example, changes during aging. So where certain species go up, certain species go down. Now, is that sufficient to induce an immune response that contributes to inflammation, which increases during aging? There's no data on that. It's only correlation. And that's why I was saying, we're at that border between what is known and then speculation. So for me, I do some speculation in the book that, the composition of the microbiome changes. So even though gut barrier function, where most of these microbes are found, gets worse, potentially leading to some of these microbes being in the blood and at higher concentrations than they usually are, it's also possible that what was in the blood to start with increased in number. And that's also the reason for why there's more of these in the blood. And then if there's more in the blood, there'll be more in the brain, which is related to Alzheimer's disease risk, because microbes have been found in the brains of Alzheimer's disease patients, or in the muscles or in the liver, all these tissues where they've been found where they're not supposed to be. So it can be both. It can be the actual composition changes leading to differences in species that can activate pro-inflammatory pathways, or it can be they're coming from the gut And that's what's causing these, pro-inflammatory pathways to impact disease and age risk. Interestingly, there are animal studies, that suggest it's only from the gut, but it's very hard to test, is it a blood story or is it a gut story? But at least in the study that I have in my mind, and that's also in the book, they actually removed the microbiome and then looked at age-related inflammation and didn't see age-related inflammation in animals that didn't have a gut microbiome. And then they also noticed the vicious cycle. So whereas, the gut microbiome triggered inflammation during aging, and then that aging triggered a worse microbiome, and vice versa, leading to a further accelerated inflammation in the body. So it's coming from a multitude of places likely. And for me, that's exciting science. There are going to be people who are studying this for the next 50 to 100 years. But just as a last note on that, it's interesting because I see so many, whether it's in the public, in the media, or, talking about Alzheimer's drugs, and this drug hasn't worked and that drug, and so many Alzheimer's disease related drugs have failed. But the idea that it's microbes is a major part of that story has been growing for 10 to 20 years. And, I bet that's going to be the dominant story. And, at some point in the future, it's just, it needs more study. So yeah, that's another reason I wrote the book, is to, put these ideas out there that they exist, and there's more than what people, know already for some of these diseases.
Yes. In Japan, people, microbes and the microbiome fascinate people all over the world, but especially in Japan, where I come from, they're truly fascinated about all those. If you go to buy a yogurt in Japan, you will see there are different kinds, and it's all about which bacteria your yogurt has, and it's not about taste, and people are really into that. But one big trend in Japan where I have seen is that microbacterial metabolites. So it's a commercial product, and people have been, I think there's an increasing interest, at least in Japan, that there has been this probiotics and those specific bacteria species you buy in a bottle or capsule or whatever it is. But more recently, I have seen a trend where companies started selling metabolites, actually, of certain bacteria, gut bacteria. The reason is because everybody cannot produce, whatever bacteria species you eat, that's only a percentage of people who can produce certain metabolites, and metabolites can be what you want. What's your take on that?
Yeah, so another great question. So there are two things there. One is that's actually the conventional opinion on optimizing the microbiome. Like, as soon as I hear or I talk about this, people will chime in with, "Oh, I eat kimchi," or "Oh, I eat yogurt or sauerkraut." But the fact is, we evolved eating fermented foods as a very small part of our evolutionary diet. For example, if a fruit fell from the tree, and it was on the floor rotting, which is fermentation, it's microbial fermentation. So to produce alcohol from the fruit sugars, that's a bacterial fermentation process. So we evolved eating some fermented products, but if you ate too much of that fermented fruit, you'd likely get drunk, and that would limit your fitness to survive. So when people talk about probiotic yogurt, I eat yogurt, and I'm sure you've seen my video on that, how it seems to impact my red blood cells.
Yeah.
And I'm not saying that's for everyone, but I'm not. And I actually did the microbiome experiment to see if it was my gut microbiome, and actually I didn't see a change, or at least in the lactobacillus, which is what I get from the yogurt. But in terms of the probiotic story, the best approach for that, which unfortunately isn't done almost by anybody, is you get your microbiome sequenced. You identify which species are actually there, whether it's bacteria, fungi, virus, etc. And then you see if you have an identified deficiency or not. And then would replacing those bacteria actually impact any of your systemic health? For example, in animal models, there's a cancer model of in animals where the mice have leukemia, and they measured their gut microbiome, and they saw that they had very low levels of a certain lactobacillus species. So they thought, all right, we're going to give that lactobacillus, and in correlation with that, they had a cancer-related muscle mass loss. So they gave micro levels of this bacteria, a lactobacillus bacteria, that was actually low in those mice. And then they replaced it to the normal levels found in the non-cancer mice. And it actually was correlated with increased muscle mass. So for me, that's the best approach. Actually measure your microbiome, see if you actually have anything that's deficient, and then do the experiment. But again, biomarkers. I mean, how do you feel on it? Is it actually optimal for your intestinal health? Do your other systemic biomarkers improve, net positive over neutral or negative? So that's the first story with the probiotics. I'd actually bet that there's a greater impact of prebiotics, which is the soluble fibers that are found in the non-starchy vegetables, because that's what's used by the gut bacteria to make some of these metabolites that you're talking about. So when bacteria convert soluble fiber, there are two types of fiber: insoluble, which is basically a bulking agent, and it makes your poo thicker, and you just poop it out, versus soluble fiber, which the bacteria in your gut will ferment into some of these quote unquote beneficial metabolites, which are short chain fatty acids. So in animal models, there are positive effects for these short chain fatty acids, which are used by the intestinal epithelial cells to actually improve gut barrier function, to keep stuff out of your blood that shouldn't be, and reduce things like inflammation, and can improve insulin sensitivity. Now, in terms of supplementing directly with these short chain fatty acids, like what if you don't want to eat a high fiber diet? You just take the short chain fatty acids and think you'll get the same effect as vegetables. Well, that could work, because there is some evidence in animal models that does work for things like muscle composition, how much fat you have in muscle, and the quantity of how much lean mass you have in aged mice. But it may not be as good as actually getting the fiber in your intestine, because when you eat some of these metabolites, they're absorbed into your small intestine and go into the blood, where they can impact your systemic health. But when you eat the soluble fiber isn't fermented, isn't absorbed into your small intestine. It goes into your large intestine. And then your large intestine, that's where it's changed into these short chain fatty acids. And then it's absorbed into the blood. So on one hand, you can probably get similar amounts of these metabolites in your blood that come from gut bacteria. But one of them is bypassing the colon, where I bet that, if you're just going to get it into your blood, you may not be optimizing your colon health, but getting it through the fiber fermentation, now you're going to optimize colon health and get those metabolites in the blood, which probably have the best bang for the buck in terms of health. Now, just further along the bacterial metabolite story, I'm actually very hot on that topic. I don't know how you probably wouldn't know that, because I haven't made that many videos in that area. But in my job as a scientist, which sounds terrible the way I just said that, but most people in the gut microbiome field, from my experience, are only looking at compositional differences. We have this, and we have this, and this is what's different in their microbiome. We have this difference in species from here. But at the end of the day, it comes down to what are they doing? And how are they doing it? And what microbes do is impacted a lot by their metabolites, their metabolism. So only saying who's there says almost nothing about how they're doing it. So in the grants that I'm submitting or have submitted this year, it's all about the metabolite story. I almost don't care about who's there, what they're doing. It's how they're doing it, which is the metabolite story. Just along those lines too, there's a scientist named Stanley Hazen who's shown that one of these bacterial metabolites, so there are good, quote unquote, good metabolites and quote unquote bad ones. So one of these metabolites is TMAO, which has some conflicting data, but at least in animal models, they've shown, and it's produced by the gut bacteria. It's actually produced into TMA, and then TMA is oxidized by our liver. So long story short is that's one of these quote unquote bad metabolites, at least in animal models. They've shown that it can impact stroke risk. Yeah. So the long story short with the metabolites is be careful which ones you're going to supplement with, because they may have positive or negative effects. The short chain fatty acids may be the best of the bunch, but there aren't very many. Actually, I can't think of any randomized clinical trials which have given these short chain fatty acids to see if they're actually beneficial for health in various health conditions. And then the issue of safety too. But that's on my list. If I can stay in science long enough, I want to, because if I have a fiber supplementation study now that's in review, and based on this premise, you eat fiber, it optimizes your gut microbiome. The gut microbiome produces more of these good metabolites, which then optimize gut function, thereby limiting the growth of bacteria that produce the quote unquote bad metabolites. And I have some of those named in the grant. But if that's true, if we directly supplement with these short chain fatty acids, which you get from the fiber, can you replicate the effects and even bypass having to eat a high fiber diet? As far as I know, there aren't any randomized controlled trials that have done that for any tissue, brain health, liver, kidney. So for people to take that based on the hope, I don't know, almost anything can be sold as a supplement, which, hopefully it isn't dangerous.
It truly can. I have to say that, it's true story. In Japan, I have seen LPS. So LPS sold as a supplement. And that's a university professor's name on the supplement, actually. And I don't know any background stories, and I haven't looked into, but I do know that such supplements exist, and that's
crazy. That's crazy. I don't know if any positive effects systemically for LPS. It activates pro-inflammatory pathways. I mean, that's
exactly. So it is true. But I'm very excited to hear your focus on metabolites, because it's so unknown, as you say. There's no study, and I keep my fingers crossed to your funding and your finding and discoveries. And I will follow your publications. I think you have 27 publications on PubMed today, and look forward to
a couple were it's Lustgarten M instead of MS, but something like that. 29, 30, something like that. It's in the ballpark, But those are studies. Those are studies that I'm proposing. Grant review takes like 6 months, and then you have to resubmit it, so it takes another 6 months. I mean, it's a long process. So, and there's no guarantee. And review, unfortunately, too. There aren't very many. The microbiome, in terms of maybe Alzheimer's disease, is much bigger than it is in muscle, which is my interest right now. So I'm being reviewed by non-microbiome specialists. And then, anyway, it's a tough time right now. I've got to work extra hard and be extra good, which is almost impossible, because I like to think I'm doing the best that I can anyway. But when your grants aren't reviewed by microbiome specialists, and the actual requests for applications aren't microbiome focused. Like if the request for an application says we want to look at microbiome and aging, those are all expired. I can't apply under those applications anymore. Now I have to go in under a general research grant, which may make it harder because there's less of an interest at the National Institute on Aging. So let's hope I can stay in science long enough to investigate these things. But I'm shooting all of my bullets in grants. I've got an exercise. Exercise too impacts the gut microbiome, and that's another story in terms of the metabolites that's almost completely unknown. Like exercise can impact the composition of the gut microbiome, who's there, but how is the gut microbiome then impacting muscle? That's known in animal models, but in people, not so much. And actually, in older people, no study. So another grant that I have currently submitted. Not reviewed, it's going to get reviewed in a few months, are the exercise-induced changes to the gut, do the gut changes actually cause changes in muscle? Nobody's done that experiment. Like if you take exercise-trained poo and put it in mice, do you get better functioning mice? So, some probiotic studies have been done like that, where it's improved function and health, but not coming from older adults that have been exercise trained. It's, young people into mice to show improvements for the gut muscle. But again, the metabolites are a part of that story in that grant. So anyway.
Yeah, they are fascinating to say the least. And as I said, I really will look forward to reading your studies and hearing about that. Dr. Lustgarten, I know that our time is almost over, and you have been very generous with your time. Any final thoughts you would like to share with people, how people can find you, or any final words you'd like to share?
Yeah, sure. I guess going to the YouTube channel would be the best place. But I'm, like I say at the end of my videos, you can find me lots of places online. I'm all over the internet, Twitter, Instagram, Facebook. It's funny because I don't know how closely you follow social media, but like names like Jake Paul and Logan Paul, do these names sound familiar?
Yes.
Yeah. So, I mean, they've got like 20 million subscribers, on YouTube, just as an example. But Jake Paul, just as a long story short, and I'll bring it back to me in a second. Jake Paul has recently, actually him and his brother have recently gone into boxing and fighting very famous people in boxing, even though they themselves are not boxers. but because they have such a large following,
Mm-hmm.
they're getting more people watching their shows than the actual fighters themselves, boxing and MMA. So anyway, it's, you know, I'm nowhere close to that level, at least not yet. But in terms of the social media, I try to be everywhere, you know, try to put out as much good quality information as possible, because I see how, you know, effective it can be in terms of educating and entertaining. So I guess the analogy I was trying to make is, you know, yeah, let's see how high this mountain goes in terms of growing the brand. So, and that's their perspective. You know, how much community engagement can we get by putting out as much information as possible? So, yeah, you can find me lots of places all over the place. Pick a social media platform, and I'm there. Yeah, thanks for having me on, Eva. This was great.
Yeah. Thank you so much for being here again, Dr. Lustgarten. This has been a true
Anytime.
treat for me today. Thank you.
Anytime.
Thank you.
This was a podcast from Kenkoax. To follow us, subscribe on the channel Kenkoax on all the places where you can find podcasts.